Hello everyone, we're going to talk about exploring the genes and the genome. First, we briefly introduce the principle of PCR. Polymerase chain reaction, which is called PCR, is a method widely used in molecular biology to rapidly make millions to billions of copies of a specific DNA sample. PCR allows scientists to take a very small sample of DNA and amplify it to a large enough amount to study in details. PCR was invented in 1983 by Carl Mullis. He awarded the Nobel Prize. The vast majority of PCR methods relies on the thermal cycling, specifically DNA melting and enzyme-driven DNA replication. PCR employs two main reagents, primers, which are short single-stranded DNA fragments known as oligonucleotides that are complementary sequence to the target DNA region, and a DNA polymerase. In the first step of PCR, the two strands of the DNA double helix are physically separated at a high temperature in a process called nucleic acid denaturation. In the second step, the temperature is lowered and the primer bound to the complementary sequence of the DNA. The two DNA strands then become the template for polymerase to enzymatically assembling a new DNA strand from free nucleotides, the building block of the DNA. As the PCR progresses, the DNA generated is itself used as a template for replication, setting in motion a chain reaction in which the original DNA template is jardinally amplified. A basic PCR setup requires several components and reagents. A DNA template that contains the DNA target region to amplify. A DNA polymerase, which is the enzyme that polymerizes new DNA strand. Two DNA primers that are complementary to the three primer ends of each of the sense and antisense strand of the DNA target. Specific primers that are complementary to the DNA target region are selected beforehand and are purchased from commercial biochemical suppliers. DNTPs, the building blocks from which the DNA polymerase synthesizes a new DNA strand. The buffer solution providing a suitable chemical environment for optimum activity and stability of the DNA polymerase, typically magnesium and manganese ions. Magnesium is the most common ion. In this slide, as shown, there are several PCR options for your purpose. So we are going to talk about the RT-PCR. Three different approaches can be used for priming cDNA reaction in two-step assays: oligo primers, random primers, or sequence-specific primers. Often, a mixture of oligo-DT and random primers is used. These primers adhere to the template mRNA strand and provide reverse transcriptase enzyme a starting point for synthesizing the cDNA. PCR primers for the co-PCR step of RT-co-PCR should ideally be designed to span an exon-exon junction, with one of the amplification primers potentially spanning the actual exon-intron boundary. This design reduces the risk of false positives from amplification of any contaminating genomic DNA, since the intron-containing genomic DNA sequence would not be amplified. RT-PCR can be performed in a one-step or two-step assay. One-step assay combines reverse transcription and PCR in a single tube and buffer, using a reverse transcriptase along with DNA polymerase. The one-step RT-PCR only utilizes sequence-specific primers. In two-step assay, The reverse transcription and PCR step are performed in separate tubes, with different optimized buffer, reaction conditions, and priming strategies. The table is the list of advantages and disadvantages when using one-step versus two-step assays in RT-PCR. After amplifying the candidate genes fragment, we move to the next step of gene cloning. Gene cloning, also known as molecular cloning, refers to the process of isolating a DNA sequence of interest for the purpose of making multiple copies of it. The identical copies are clones to make recombinant DNA, a form of artificial DNA. Gene cloning amplifies a specific piece of DNA via bacterial cells. To do gene clone, we need clone vectors and competent bacterial cells. Cloning vector, which is a replication DNA molecules attached with a foreign DNA fragment to be introduced into a competent cell. There are several commercialized cloning vectors. An idealized cloning vector has the origin of a replication, one or more selectable markers, and one or more unique restriction sites for enzyme to cut. Application of gene clone: 1. Genome organization and gene expression. 2nd, they could be used as production of recombinant proteins to get enough amount of candidate proteins. And also, we could do the transgenic algorithms or do the gene therapy. Here is a summary of gene cloning process. First, we use a restriction enzyme, which is a protein that recognizes a specific short nucleotide sequence and cut the DNA only at that specific site, which is known as the restriction site to target sequence. More than 400 restriction enzymes have been isolated from the bacteria. Step of the gene clone: choice of the algorithm and the clone vector. We need to prepare of the vector DNA, preparation of DNA to be cloned, and creating the recombinant DNA with a DNA ligase. And also we need to introduce the recombinant DNA into the host organisms. And after that, we do the selections of the algorithm which contain vector sequence, screening for clone with desired DNA insert and biological properties. So the ligase gene can be used to screen bacteria containing recombinant plasmid, which is a special plasmid carrying a copy of the ligase genes and amphithenin-resistant genes. Then move to the plasmid extraction. Connect the appropriate antibiotic with a single colony of the transformed bacteria and incubate the culture overnight at 37 degrees with vigorous shaking. Then centrifuge at the maximum speed for 30 seconds to connect the bacterial pellet as dry as possible. And then after that, we lysate the bacterial suspensions and purify the plasmid by filtering through the membrane. Dissolve the nucleotides in 50 microliters of the tea buffer. After that, we measure the purity of the plasmid by optical density machine or axial electrophoresis. The accuracy of the plasmid will be verified via single-section analysis or restriction site cut. Broadly defined, transfection is the process of the artificial introducing nucleotides, DNA or RNA into the cells. Such introduction of foreign nucleotides using various chemical, biological, or physical methods can result in a change of property of the cells, allowing the study of gene function and protein expression in the context of the cell. We use lipid-mediated gene delivery example. It uses lipids to cause a cell to absorb exogenous DNA, since they are both made of the phospholipid bilayer. We will discuss the gene expression in cells in next lecture. And this is the end of this lecture. Here's some reference, and thank you for your attention.